Okay, welcome friends. Welcome to AcadHelp BioWebinar.
Yes.
Now, those who are new to the series, let me tell you about this program. This BioWebinar is a series of web series where selected eminent teachers are invited to talk and discuss on specific areas in biology. We discuss various chapters of biology of senior secondary and secondary level. And our objective is to enrich the students, to give them more information and enrich them, and especially to give them those answers which have remained unanswered to them. So in this line, we are working together with our eminent teachers, and we hope that our program will be successful and our students will be benefited. So now let me introduce you our panelists today. Now we have Mr. Gautam Biswas on the left, right top. He is a postgraduate teacher in biology, now working in Kendriya Vidyalaya, Andal in West Bengal. He has more than 20 years of experience in teaching. Next is Dr. Sabyasachi Majumdar, an eminent teacher. He's a national teacher. He has more than 25 years of teaching experience. And we have with us a young, energetic postgraduate teacher, Mr. Rajiv Ranjan Singh. He's from Kendriya Vidyalaya number 1, Kumbhigram, which is in Kachar district of Assam. So welcome the guests and welcome the viewers. Today's discussion is continuation of the previous discussions, previous webinar I can say, that is human reproduction. So this is the second part, human reproduction part 2 now. Here we'll be discussing certain areas one by one. We'll go to the panelists for their expert opinions. We have questions for them also, and these questions are actually sent by the students. And we'll ask them also. They will give us the correct answer for this, and students will be enjoying and benefiting it. So once again, before I start, I'd like to give a grand welcome to all the 3 experts.
Thank you, sir.
To start with, our first discussion will be on spermatogenesis. Our series last ended with oogenesis, and we'll start with spermatogenesis. And I'd like to ask Dr. Majumdar. Dr. Majumdar,
yes,
I'd like you to throw some light on spermatogenesis, because this is an area where students very oftenly they ask questions and they confuse also. So please elaborate this. Spermatogenesis, where it occurs, when it occurs, and how it occurs. Please.
So, dear friends, you have just raised a pertinent question, pertinent topic, spermatogenesis. On the other day, my friend Rajiv has given an account of the oogenesis. This, in all sexually reproducing organisms, gametes are very important. And so formation of gamete, in one word we can call gametogenesis, and male gametogenesis comes under spermatogenesis. If we elaborate a bit, that is formation of haploid, functional, motile spermatozoa.
Okay.
From diploid primordial germ cells of the seminiferous tubules of the testis can comes under spermatogenesis. Okay, and one more important thing is that gametes are haploid in nature to maintain the constancy of chromosome number. This is very important. And another important thing is that since there is meiosis, so So the genetic variability is also, it happens here through crossing over. So these are important things that happens in both in spermatogenesis and oogenesis. Now I'm discussing certain points on this. Okay.
Yes.
So that's spermatogenesis. So at the beginning, what we can call it, that the primordial germ cells, those progenitor cells of the stem cells, there are type A cells, two types of type A cells. That is the dark cells and the pale cells. These dark type A cells, they usually are very important to maintain the number of these primordial germ cells. They go on dividing mitotically and producing these germ cells only. Whereas the pale cells, type A cells, they undergoes mitosis to give rise to type B cells.
Dr. Majumdar, here I'd like to ask one question. This dark cells and pale cells which you are talking of, is it because of the intensity of the color of the nuclei?
Color of the nucleus. Color of the nucleus based on.
Okay.
And another important thing is that these type B cells ultimately are responsible for paving the way for production of sperms, spermatozoa. So that's most important. In our previous book, there were some light on it, but in the new book, it is not been mentioned. But this is very important, and this is part of the spermatocytogenesis that is important. The nomenclature is also important, as because in biology, naming is very important. And here is sperma, that is seed, and genesis, that is origin of seed, male seeds, that is gametes, spermatogenesis. And it takes about 65 to 70 days for sperm production. Okay. And if you go through this slide, you'll find that there are three phases. The multiplication phase, where these stem cells, they undergoes mitotic, rapid proliferation, mitotic division. That is the multiplication phase. And each of these type B spermatogonia undergoes growth. That is the growth phase. And during this growth phase, they undergo certain changes. So this happens, and then immediately after the production of this primary spermatocyte, the DNA replication takes place. Now the point is that this primary spermatocyte is basically a myocyte. Students very often fail to correlate this because these two are in different pages of the book. They try to cram about myocyte, but it is very easy if we can correlate here that the cells that undergoes meiosis is myocyte. So in case of this, primary spermatocyte is the myocyte.
Okay.
This primary spermatocyte, yes, anything?
No, okay.
Primary spermatocyte undergoes meiosis. First meiotic division, that is basically a
reduction division.
And each of these primary spermatocyte give rise to, here you see, give rise to two secondary spermatocytes. Primary spermatocytes, as I've told you, are diploid in nature. The students must mention diploid and within bracket the ploidy number 2n. And it gives rise to secondary spermatocyte, which is n, because of the first meiotic division. And here one point is very important, that it undergoes equal cytoplasmic division, unlike that of the oogenesis, where we get unequal cytoplasmic division leading to one secondary oocyte and one polar body. Here it is, both are secondary spermatocytes.
Okay.
And the secondary spermatocyte undergoes the second meiotic division to give rise to, each of these secondary spermatocyte will give rise to 2 spermatids. So from one primary spermatocyte, actually we are getting 4 haploid spermatids.
Okay, Dr. Majumdar, I'd like to have one question for you.
Yes.
Because while discussing this, I just received a question from some students asking that there are 3 different terms which are very confusing while studying spermatogenesis. One is spermatogenesis, second is spermiation, and third is spermiogenesis.
Yes, yes, I will be coming.
Okay, please, during your discussion, please elaborate this part also.
Yes, 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 yes. Now, you see, these spermatids, here you see, in this particular diagram, you can notice that there is cytoplasmic connection, cytoplasmic bridge. That is very important. In case of spermatogenesis, they remain cytoplasmically attached. As because, you know, that we are having X and Y bearing gametes. And especially the Y bearing gametes, they are lacking in some X bearing proteins because of lack of genes. So,
okay, Majumdar, please restrict to this diagram because I think students will not be able to.
So that is important. Cytoplasmic bridge is there. It remains to spermatid formation.
Okay, okay.
Spermatids undergoes transformation. Here you see.
Okay, okay.
This transformation, differentiation of spermatids.
Yeah, this is a wonderful diagram.
And there are a lot of changes takes place. First of all, the nucleus condensed, the DNA condensed. It takes a position in the head region. The Golgi vesicles forms a chromosomal cap over the nucleus, and that contains the lytic enzyme sperm lysine hyaluronidase. You can see these centrioles, they play a vital role in their tail formation, flagella formation, axoneme, especially the distal centrioles, microtubule formation, shading of cytoplasm. That is another important thing, very important thing, which is ultimately engulfed by the Sertoli cells. The mitochondria, they become arranged around the midpiece as mitochondrial cloud. Very important features, because the motility of the sperm depends upon huge energy is needed, and that is why it is mitochondria cloud is there. And that, as I've told you, that this sperm flagellum, that's the axoneme or the microtubular scaffolding, and the protein structure, flagellin and dynein, these are also being produced during this time. And that is why the motility still they get the tail piece. So the head, the mid piece, then the tail piece with this acrosome, nucleus, the centrioles, proximal and the distal centriole, and the tail piece. Middle piece with mitochondria and the tail piece. So that's very important changes, and this comes under spermiogenesis or spermatogenesis. And students often confuse this with spermiation. So spermatogenesis or spermiogenesis is basically a part of the spermatogenesis, the last phase of spermatogenesis, in which the non-motile spermatids, non-motile haploid spermatids is transformed into functional motile spermatozoa. So that's one terminology, and they are still embedded within the Satori cells, and gradually they loses because of that nutritive function, and gradually they get detached from the Satori cells and comes to the lumen, and then it is pushes towards the ducts, as we have discussed yesterday on the other day, and this comes under spermiation process. So that is spermatogenesis, spermacytogenesis, spermiation, and spermiogenesis, the different terms.
Thank you, Dr. Sumisachi Majumdar. I suppose now it is very clear that these three important terms, that is spermatogenesis, which is only, which you can very simply say it as formation of sperms, and or the various steps which involves in the formation of sperms is spermatogenesis. Next term that which you have just pointed out is spermiogenesis, that is transformation of spermatid into the typical tadpole-shaped spermatozoa. Am I right?
Yes, yes.
And the last part, is
so the ploidy number remains same.
Yes, and the last part, that is the spermiation, which means that the sperm cells which were embedded in the Sertoli cells, they are finally released into the seminiferous tubules. So this process of release of the spermatozoa is known as spermiation. This is what I suppose you wanted to say, and this is the gist of the three terms which you have used. Now may I ask Mr. Gautam Biswas?
Yes, yes.
The structure of the sperm.
Hello?
Hello.
Yes.
Can you share the structure of the sperm?
Structure of the sperm. Just a minute, sir. Let me see.
Or Rajiv, can you? Anyone? Okay, here already there is a structure. This can be elaborated, no problem. Okay, already present on the screen. Already it is present on the screen. Okay, so here as we find that the sperm has the head region, neck region, middle piece, and the tail, right? The head portion has acrosome, that's the tip portion, and the nucleus. And then there is the centrioles, and then the mitochondria in the middle piece, and the long tail. So in brief, this is the structure of the spermatozoa. Okay, now thank you, Dr. Majumdar. Now let's come to Gautam, Mr. Gautam.
Yes.
Now I'd like you to, okay, but before coming to you, I think I must ask Mr. Rajiv to speak on fertilization. Mr. Rajiv, fertilization. Please explain this process of fertilization. There is a lot of confusion in this. No sound, no sound. Sound, please. Rajiv, sound. Sound problem. Rajiv, sound.
Yeah, hello. Am I audible?
Yes.
Repeat from the first slide.
Sure. So when we talk about fertilization, it is a set of events. Basically, fusion of male and female gamete comes under preview of fertilization, but it needs certain preparation, certain synchronization, and at end, when there is a fusion of male and female gamete, we talk that event as fertilization, right? So as we can see, let us start from the structure of the both gametes. When at ovulation, we call generally it as ovum, but at this stage it is nothing but secondary oocyte, right? Along with some follicular cells as corona radiata. And as you have also described about the spermatozoa structure and the significance of different part. So we can just see that what are the events. We can list them one by one. Yes, so what are the events? First of all, ovum needs to arrive at the site of fertilization. The site of fertilization happens to be the oviduct and specifically the ampulla asthenic junction,
Yes.
right?
Yes, yes.
So after ovulation, the ovum comes out through ostia, taken into oviducal funnel and moves passively to the ampulla asthenic junction. And this takes some time. It takes around 8 to 12 hours because it's important for ovum to reach that place.
Yes.
Then it has to be synchronized with the insemination. That is deposition of semen. And we know that semen contains huge number of spermatozoa, 80 to 100 million per ml. And as it is deposited in the female genital tract, it forms a coagulum. And it gets freed after around 15-20 minutes so that it starts ascending and gliding. And in that, the motility of sperm is boosted by certain other factors. Sperm starts swimming actively, and the rate is around 1.5 to 3 millimeter per minute. So effectively to reach the site of fertilization, it takes around 6 to 8 hours. 6 hours, right? And as Dr. Sabyasachi has already explained, the composition of semen, the prostaglandin in the semen, even the oxytocin released from the female pituitary and cervical mucus, all this together help in vigorous movement of the female reproductive tract as well as ascending and gliding of the sperm. At this point, sperm does not have the capability to fertilize. Why? Because in semen there are certain factors which we call as decapacitation factor, right? So what is to be done now? The sperm has to be capacitated. So in the female reproductive tract, the sperm's decapacitation factors are diluted. The inhibitory factors are diluted. There is a thin membrane of cholesterol over the acrosome, right? That is also removed. And the permeability of acrosome increases and sperm also imbibes calcium ion, right? This all ensures that now it is ready to fertilize and fusion of gamete is approaching. At this stage, we can also list there are certain sub-phases that we can talk about as discrete events. First of all, we talk about acrosomal reaction. As Dr. Sabyasachi has explained that acrosome contains sperm lysine, hyaluronidase. So what is the function of hyaluronidase? It dissolves the cementing material of corona cells. There is another enzyme, corona penetrating enzyme, that degenerates the obstructing corona cells, right? And now when the sperm is approaching the ovum membrane, what happens at zona pellucida? There is a typical reaction that takes place, compatibility check. We talk about species-specific fertilization, right? It is needed. So what factor? It is here only that at zona pellucida there are certain receptors. In egg, it is called as fertilizing and anti-fertilizing in sperm. And when there is a compatibility, then sperm acrosin is released and that degrades the zona pellucida.
I think it is for this reason that fertilization is called as a physicochemical event.
True.
And it is species specific.
And it is species
Specific and physiochemical process.
Fertilizing. Okay, please continue.
And the physiological factors here out, the pH remains around 7.4 and it needs calcium and magnesium ion as well. Now we look at sperm entry. What part of sperm enters? That's another area. It is the sperm head, which comes in contact with the fertilization cone, because whenever the zona pellucida degrades, the oolemma protrudes from there, forming a fertilization cone, right? So, and through that cone, at that point, the sperm and egg membrane dissolves, and thereby some amount of calcium is pushed in the ovum. So here also, you know, just like a potential difference, a potential change takes place. So sperm's component of head, middle piece and neck, that enters the sperm, and tail is left out because tail's function is propulsion.
Okay.
Then comes very important reaction, that is called as cortical reaction. The calcium ion, what it does, the calcium ion causes extrusion of cortical granules from the egg, and that converts the plasma membrane into activated calyx. So now plasma membrane will be
at this time when there is a sperm contact, there is a depolarization in the membrane.
Exactly, depolarization. That's why I said a potential gradient is developed. So now the cortical grain extrudes, it converts the plasma membrane into fertilization membrane. So this is one factor that stops polyspermy. There is no second chance for other sperm to enter.
I suppose this is a very important point which is being discussed, and is very often asked why the second sperm do not enter or is not allowed to enter into the egg.
But it is not only the cortical reaction. Another set of reaction called as Jonah reaction, that also helps in the same. The chemical that egg extrude, that reaches Jonah pellucida and destroys the sperm receptor. So beautiful mechanism.
Okay, this is very wonderful. So there are two factors which we find, that is cortical reaction and Jonah reaction.
Jonah reaction
are the which are the main reason why there is no extra or the second sperm fertilizing the ovum. That's good. Fine. Fantastic. Yes, please continue.
Right. So now the impulse has to go to the secondary oocyte. I repeat again, not ovum, secondary oocyte, because oogenesis has not completed yet. So the semen contains, the sperm contains, the component of sperm contains PCL zeta protein, right? Which gives a signal to the arrested oocyte, right? And it removes the metaphase promoting factor and develops anaphase promoting complex. Very, very important. So this gives the signal to the secondary oocyte to complete meiosis 2. And as a result,
I have a question for you now. Now see, what you have just said, it has it will be sufficient enough to confuse a student, because in the other day we had discussed that oogenesis starts in the fetal age when the girl child is in the mother's womb, means fetal stage. And now the oogenesis started at that time and it is ending when there is fertilization. So this is a very long process.
Exactly, it is long process, but keeping the...
Compared to what Dr. Majumdar had just said, the spermatogenesis takes about 60 to 65 hours. And this takes so many years. Huh?
No, see, one thing has to be kept in mind. The ovum, which contains the cytoplasm and other factors needed for further embryonic development. So that is one, it is unique, it has to be preserved, protected, and economization of resources is another principle that biological system follows. That's why it takes long, unless, because at this point, when sperm reaches, there can be two possibilities. Either fertilization will take place or it will not take place. So only when the fertilization will take place, then only we need the ovum. So it is need-based progression. Now when it is sure that sperm has entered, it has approached, now secondary oocyte will convert it into ovum and second polar body. Right?
Okay.
And then comes the term that we have been talking about, the fertilization. The male and female pronucleus comes closer. The membrane gets dissolved and paternal and maternal chromosomes that mixes and forms a zygote, and the zygote is diploid. What Dr. Sabyasachi has told, restoration of chromosome constant is one of the very important factor for any organism.
Continuation of the species.
Yes, this is what.
Okay.
And a student can take one more slide. This one, I guess, where we give a complete series, what is happening. Sperm binding receptors are beautifully shown, and how acrosome is releasing content. There is a compatibility, that then dissolution of, you see here, how dissolution of the membrane, fertilization cone formation, content is getting out, right? So overall, this is a beautiful process.
This is, I suppose, a fantastic diagram where you can see the stepwise the events which is happening with the arrival of the sperm, coming in contact with the membrane, and then releasing hydrolytic enzymes. And due to this, the receptors on the membrane, you see they are being activated. And due to this activation, there is again cortical reaction. And in the perivitalian space, the cortical granules are releasing their content. That's fantastic. Fine. It's a wonderful diagram. And it is sufficient for a student to understand what you wanted actually to say. Your explanation,
because this step is going to make two things clear.
Diagram is also wonderful. Yes.
Thank you.
Okay, that was a wonderful presentation. And now let's go to Mr. Gautam. Mr. Gautam Biswas.
Yes.
So by this time we have understood, or we have seen how these things are formed. The gametes are formed, right?
Yes.
That is formation of the ovum, I should say the secondary oocyte, and also the formation of the sperms. And
Yes.
also by this time we have understood the process of fertilization, the events which are
Yes.
taking place. Now we
Yes.
go to the next step, one step ahead of this. What
Okay.
happens? Now just now Mr. Rajiv has explained that
Yes.
fertilization takes place in the ampulla isthmic region.
Yeah.
After this, what happens?
Well,
If there is no fertilization, what will happen? We'll discuss later on. But now let us
okay.
discuss with what will happen if there is fertilization. What will be the next steps? Please.
Okay. Sure. Sure.
Mr. Biswas.
Yes. Sure. Just a minute. Now, let's come towards after fertilization. So what we have seen, the last case was, I think the screen is visible.
Yeah.
The fertilization event which you have seen, shown well by Rajiv in very detail. So the first thing which comes over after fertilization is the first diploid cell of life.
Right.
Okay. So the first diploid cell of life, that is the zygote.
Zygote.
And this takes place where? Obviously in the fallopian tube. Okay, that's the ampulla-isthmic zone. And then it starts proceeding towards the uterus. By the time all other divisions will be mitosis. So the meiotic division had already been completed during gamete formation. Now no other further meiotic division will take place. Now during this mitotic division, these mitotic divisions are rather called as cleavage division. Okay, repeated mitotic division, we can call it as cleavage division. And due to this,
you're
yes,
saying that it is a mitotic division, but you want to term
yes,
it as separate as a cleavage division. Why?
yes, means the process of division, the chromosome number will now remain same in all the cells.
Right.
So the division will make from 1 cell to 2, 2 to 4, 4 to 8, 8 to 16, and so on. The number of cells will go on. But
how is
keeping
it different from mitosis?
Yes.
How is it different from mitosis?
Well, over here, what happens in case of mitosis after division? Say there will be differentiation or increase, but over here the number of cell increases, the size do not increase because zona pellucida layer still remains intact.
Okay, that means you want to say that in mitosis, after the formation of two daughter cells, the two daughter cells grow in size.
Yes.
G1 is G2 complete,
Yes.
and then again they go into mitosis for the next division.
Yes, exactly.
So there is
exactly.
growth of the cells, but here in cleavage division there is no growth of the cells.
Okay.
Or here the blastomeres.
Yes,
right?
yes.
And that is why there is a progressive reduction in the size of the daughter cells.
But overall increase in number of cells.
Yes, increase in the number of cells, but
increase
reduction
in number of cells, but reduction in size of cells.
Okay, right.
Okay.
This is the main difference between cleavage and mitosis.
Yes. Okay. So the thing is, we can just now we can see the series from zygote.
Right.
Okay. Cleavage
So
division.
We have studied up to zygote, means the fertilization, which I think just now Rajiv has explained.
Yes.
Okay.
So due to cleavage division, initially a ball-like structure forms a solid ball, which is called as morula, which means little mulberry.
Right.
So here the cells are compact without any space inside it. If you compare this one, I can give a homology between this one morula and the next step blastula, which has a hollow space inside. We can just compare with a cricket ball, solid cricket ball, and a tennis ball. If you cut a solid cricket ball, you won't find any space inside. But if you cut a tennis ball, you will find a space inside. So blastula is somewhat like that of tennis ball, but has a difference. We'll come to it. Still, it has a difference. That is, it has a mass of cell in one end. Okay. And next, after blastula, it gets implanted. The remaining part will be gastrulation and organogenesis. Let's see the slide.
Okay, then
and see over here. Okay, what happens? So we can see this is ovulation. Consider that this is day 14.
Right.
Okay. So it reaches over here, say within 12 to 24 hours. So by day 15, they get fertilized. and say the day of fertilization, if you take the day of fertilization as day 0, then after that the division starts and it progresses and it comes to the endometrium in the uterus and gets
Implant.
embedded in it, which we call as implantation.
Right.
The total time period is taken nearly 7 days. Within 7 days of after fertilization, if it is not implanted, then this blastula will be destroyed. Okay, I'm coming to this one. Suppose we this one I'm showing
Okay,
inside the fallopian tube.
Mr.
If
Mr.
we
Biswas,
just see the diagram in this way.
Mr. Biswas, I have a
Yes.
question for you. Now,
Yes.
just now you said that if there is no implantation within 7 or 8 days, whatever it is, then it
Yes.
is destroyed. Then should we call it as a miscarriage? Will it be then miscarriage?
No, it should not be. It should not be called as miscarriage in such cases, because implantation miscarriage is actually having some more time. I think so. Some, if you think about the pregnancy, then it will take some more time after implantation. If the implantation is removed off, then we can say it is miscarriage. But here implantation has not taken place yet.
Okay, okay, okay.
So this is a failure of implantation, rather you can say.
Okay, okay, okay.
Okay, so we will
This
state
also-
over here as
this also
miscarriage.
can occur in certain females, and that will also
Yes.
be a problem with the bearing a child. Even in
Yeah.
spite of being fertilized,
Yes.
still due to lack of this ability to implant,
Yes.
the woman will not be able to bear a child. So there are various factors, I suppose, now.
Yes, you just referred to now. We can, when we go to next chapter, chapter 4 in assisted reproductive techniques, there we'll find that there can be several causes how a couple may not get a child, or there can be problem in conceiving.
Right, right, right.
So
We'll
in that case we can
chapter
discuss
later on. Okay, please.
reproductive health.
Right, please continue.
Okay.
Please continue.
So here it has been shown the diagrams. If you see over here, that zygote, of course, this was said. Now the first cleavage division forming 2 cells. Okay. Next, the second cleavage division is a right angle to the first one, forming 4 cells.
First
Third one is right angle to the
first division. I think it is along the
Yes.
animal vegetal pole.
Yes. It's through the animal vegetal pole, and a complete division.
Okay.
So holoblastic cleavage.
Okay, right.
Okay. And in some cases, other animals, we also have meroblastic cleavage. I'm not going over there because
No, we don't want
that's
to go
not
to
included in
details.
NCERT text. So I'm trying to keep my lecture as
Okay.
far as possible with respect to the CBSE or the NCERT content and for the students.
Okay.
So the next division is again right angle to the first division, and in this way they form morula, early morula and late morula, which is like a solid ball. So
Right.
this by day 4 we find the morula, and next by day 5, 6 we get blastula, where a inner space develops. We'll come to the structure of blastula and then again implantation by this diagram. Okay, now you see the blastula over here. This is the diagram I have given over here too. The thing is same only, but have a difference. Now this blastula is made up of 2 layers. One is called as tropoblast, another is inner cell mass. This inner cell mass is basically the germ cells, which will actually form the embryo proper.
Okay.
And the tropoblast, the word tropo means feeding. So it helps in feeding the growing fetus next, or the germ cells. So this will develop into placenta.
Okay.
And also it will also help in releasing certain hormones. So let's see the thing over here. What is said, if embryo must implant into the wall of the uterus within 7 days. Mind it should get implanted within 7 days, or it will deteriorate and die. We will not call this as miscarriage. Okay.
Okay.
Next, blastocyst or the blastula gets embedded in the endometrium lining, leading to pregnancy. So we'll only say the lady or the female is pregnant if the blastocyst gets embedded in the endometrium. Otherwise, she is not pregnant.
Okay. Okay.
Okay. Next, the blastocyst, over where we find the arrangement of cells, as I have said. Now you try to observe these 2 diagrams. In this 2 diagram, you see over here you have a layer saying degrading zona pellucida, which is absent over here.
Yes.
Okay, so this is prior to implantation. During implantation, this zona pellucida layer is there, then there will be no implantation.
Yeah,
because
implantation,
this
they have
tropoderm,
contact with the trophoblast.
yes, trophoblast, they will
contact.
form the finger-like projections to form the villi
Yes.
and to for further development of the placenta. Okay, so there's this is another again part when the tropoderm it forms the placenta.
During implantation, this
Yes.
inner cell mass region, it forms the embryonal knob.
Yes.
This part is actually implanted,
Yes.
and the trophoblast, they release some enzymes. These enzymes
yes.
degrades the endometrial wall and creates a pit. And
exactly.
and the blastula gets impregnated into the pit.
Yes. Yes. At the same time, this trophoblast also releases human chorionic gonadotropin.
Oh, that's very important.
And this is the test for pregnancy positive is actually detection of human chorionic gonadotropin in the urine. Okay. So if this hormone is present, then we can detect that yes, pregnancy have taken place. If it is absent, we say no, it had not taken place. Well,
okay.
This is how the implantation takes place. Okay.
Okay.
You can see over here the process. It is going inside the endometrium. Okay.
Right.
So the trophoblast, it gives rise to the placenta, and this placenta is an interface. The question rises, placenta is whose property? Either mother or the fetus whose? It is actually maternal and fetal interface. It's property of both. Okay. So by this, the placentation takes place and further division will be gastrulation.
Okay. We'll continue.
We'll come to it. Yes.
We'll continue with the discussion because there are a lot more to be discussed.
Yes.
This is just the second part of this chapter which we have just discussed. And we are coming to an end of today's discussion.
Okay.
And today we have discussed at length 3 very, very important areas of the chapter, human reproduction. And I'd now like to thank our guests. They have done a wonderful job. They have explained very much in detail. So I thank Rajiv Ranjan Singh, his explanation with wonderful diagrams, and also Mr. Gautam Biswas, wonderfully explained with supportive diagrams, and also Dr. Majumdar. So thanks a lot, and thanks to the viewers.
Thank you.
We'll meet again in the same chapter, Human Reproduction, Part 3. Thank you very much.
Thank you.
Thank you.